snakes, fear of heights, large predators like a lion, or small spaces. These are all really important things to fear because they will kill you. So when we encounter things that make us afraid, we have a facial expression. So I want to talk about how faces are important first. Faces are important in an evolutionary point of view because we are a social species and we rely on groups, our social groups, to survive. So it's important to be able to recognize other people's faces, and also that comes with it emotions. Emotions can tell you a lot about the environment that you're in. So one of the first people that I know to study facial expressions was Duchenne. How many people have heard of Duchenne's smile? A small handful of people. Okay, so the Duchenne smile means genuine smile. Basically what he did is I'm going to take this off real quick. So basically what Duchenne did is he electrically stimulated muscles in the face. In this example, he's stimulating the zygomatic major. And that's a muscle that you use to just have a normal, like when you smile for a picture and you can tell that it's not really a genuine smile, and you're just like, hey, I'm here. But a genuine smile involves the orbicularis oculi. I hope I'm saying that right to anyone that knows what that is. And that's the muscle around the eye. So when you have a genuine smile, that is also contracted. The other thing Duchenne noticed was with fearful facial expressions, those are also hard to voluntarily produce. Now it looks like he's kind of torturing this poor guy, but he was a neurologist in France he published this work in 1862. This gentleman here, his patient, has near total facial anesthesia, so he can't really move his facial muscles to begin with, so this was a good person to study. Darwin was interested in Duchenne's research and published on facial expressions as well. And one of Darwin's questions was, does facial expressions have a biological basis? And he hypothesized that if people from other cultures have the same facial expressions and meanings, then there must be a biological basis and not a culturally learned expression. So what he did is he asked individuals, Englishmen that are in other countries or traveling to other countries, to document the facial expressions and their meanings, and their reports suggested that supported his hypothesis. However, this had a lot of controversy because he didn't actually experiment anything, it was just observation, and he's just basing everything off of these random Englishmen's opinions. So there's could be a lot of confounds in this research. However, Erickson studied the 4 people in New Guinea. They were secluded from the Western world up until 1958. And what he did with them was he read an emotional story in their native language, so like a sad story or a happy story, and then had them identify the facial expression that could correlate with that story. And what they found was they identified the correct story, and I say the correct emotional face of the story. I say correct because if it was different, then it's not wrong, it's just different. So this indicates that there could be a biological basis for facial expressions. Now, why is a fearful face, why do we have our eyes wide open and our mouth open and our nostrils a little more open? There's functionality reasons for it. When you have a fearful face, your eyes are open, you have greater visual field, you can move your eyes faster, you have greater air volume intake, so that if you need to run, you can run. And that's comparing a neutral face. And then you can also compare neutral to disgusted face, where everything's all squished up and puckered because something's disgusting. You don't really want to look at it or smell it. So now it's time for the straws. So if you could unwrap your straws, I'll give you a little background so you don't have to keep it in your mouth for a long period of time. But facial feedback hypothesis. So what does your facial expression tell your own brain? So basically, people used to study this hypothesis by electrically stimulating the facial muscles like Duchenne did. However, that's uncomfortable, so if you're trying to make someone smile and see if they'll think something's more funny, then it might not be more funny because your face has electricity going through it. So what they decided is to use a pen. I brought straws, because I don't want to buy everyone a pen. So if everyone could hold the pen in their mouth, like this person on your right, use your teeth, so that way it kind of forces a smile, and I know some people say that you can frown while holding with your teeth, but don't. It's not the point. So then they showed them cartoons like this, and they would ask the people to rate if the funniness and amusement level of these cartoons. And what they found is that people that held it in the teeth, like you guys, found it more funny than the control group that held a pen in their hand. And the control group found it more funny than the people that held it with their lip that kind of made like a sad puckered face. So, according to science, keep it in your mouth and make my job easier, because I'll be more funny and more amusing if you keep forcing yourself to smile. And don't worry, the reason I picked straws is so you can continue drinking your beer. Or water. So now let's talk about the brain. Ledoux proposed a high road and low road theory of fear response. So when something comes into your visual field, whether you know that it's there or not, if something comes into your visual field, the information from your retina goes to your thalamus and then to the amygdala. That is the low road, or also we call it the quick and dirty route, because it's not really detailed and it's a very fuzzy image of what something is. And the amygdala will determine if it's something that resembles something that you should be afraid of. So in this case, it looks kind of like something with large fangs, so maybe we should be scared of it, go run the other way, or whatever was the best option to do. However, there's the high road, where information from the retina goes to the thalamus, to the visual cortex, gets further processed, so that way you can actually be aware of what you are seeing and actually be able to know what it is. In this case, you might not need to be afraid of it because it's just a little kitten meowing. Now I know that's not really like a real world example because you're not going to mistake a little kitten for a tiger. I hope not. If you do, talk to someone. But another example is if you're walking on a trail, which I hope you live in Marquette, so hopefully you do, because you have so many trails to walk down. And you're just walking along, you look at the trees, and you're looking at the sky, and then all of a sudden you kind of jump back and move over here, and you look down and you see a stick, and you're like, oh, it's just a stick, and you keep walking. What happened was the amygdala thought it saw a snake, so it made you jump back. And then the high road continued further processing and realized, oh, it's just a stick, it's fine, and then you return back to normal, your heart rate calms down, and you calm down. And it's important to be able to do that because that split second is crucial. If that was a diamondback rattlesnake, you're going to want to jump back before you actually process what it was, because those few milliseconds could mean life or death. And also, another example could be a moving car. Your amygdala could actually be activated by a moving car that you are not aware of. And we'll talk about awareness in a second. But how important is the amygdala in fear response? Well, patient SM had reduced experience of fear because she didn't have amygdala. Both of her amygdalae were degenerated through Urbach-Weiss disease. You can see here, this is where the amygdala is located. This is a coronal slice, so it's right here. You're looking at the face is cut off. So she had a reduced experience of fear. Not that she didn't understand the concept of fear, but she knew that she didn't like snakes, and she would say that she's afraid of snakes. But then researchers gave her a snake, and she was fine holding it, and there was no problem. And there's some other stories with her that she was robbed at gunpoint, and then she almost got hit by a car, because she doesn't have this initial fear response that we all rely on every day. So what researchers wanted to do was they had her draw facial expressions, happy, sad, and fearful. And what she ended up doing but she drew them all correct, except she drew a little baby with an angry face and hair standing up for afraid. Now, the researchers were curious about that and asked her, why did you draw that? When the other ones, these are pretty good sketches, I couldn't even do them. She said that she's thought babies are always scared because they're always crying, and she knows that when you're scared, the hair stands up on the back of your neck. So therefore, that is afraid. Now, that made the researchers more interested in, can she identify emotional faces? So they used an eye tracking device and asked her to identify emotional expressions. This is the control group. These little marks are where they looked. And as you can see, the control group looked at the eyes for all the expressions, and the red bar right here, that's the control group. They all did pretty well at identifying what the facial expressions were. And SM also did a pretty good job identifying all the facial expressions except for fear. And then if you look at the fearful face, she stared at the nose the whole time. But then when they instructed her to look at the eyes, then she was able to know that that was a fearful face. So we have an attentional bias towards threat, and we also have an attentional bias towards fearful faces, because a fearful face will tell you that there is a threat in your environment. And the amygdala is also activated during this time, or during when you attend to a fearful face. And one way that we can test this is through a dot probe task. Basically, it starts with a fixation cue in the center of the screen, and then two faces on either side of the screen, a fearful and a neutral, and then a dot. The participants asked to just locate which side of the screen the dot showed up on. That's all they have to do. For this example, because it appeared behind the fearful face, that's a congruent trial, because your attention is typically oriented towards the fearful face, and your reaction time will be pretty quick at that dot because your attention's already there. And then we have another condition another trial type that's incongruent. So you have your neutral and fearful, and the dot appears on the other side of the screen. That typically takes longer to respond to because you have to reorient your attention from that side of the screen to this side. We also have a baseline trial where it's two neutral faces, so your attention doesn't really be directed in any way or orient or either side. The dot appears, you react to it, and that's kind of the baseline of how long it takes to just react to a dot. We also have a method of backward masking. So we show these two faces for 33 milliseconds, so really quick, and then we replace them with two neutral faces. And for the most part, most people aren't aware that they even saw a fearful face. And I'll get into that more later too. And then again, this would be an example of a congruent trial. So a model for an attentional bias towards fearful faces is very similar to Ledoux's high road and low road, except this feedback system right here from the prefrontal cortex to the amygdala. And we think that that's involved, I say think because it needs further research, like everything else. We think that's involved with reorienting attention control, because the prefrontal cortex is typically involved in cognitive function like attention. So what do we do in Cabin Lab? Cabin Lab is a cognitive by affective behavior integrated neuroscience lab headed by Dr. Joshua Carlson right here. So what do we do besides procrastinate? ACPC practice. Hopefully one day you guys will know what that is. That's our band. And also for me, I came up with a new thesis topic every 10 minutes. Where I finally figured it out. So one of the first questions we asked is, how important are the eyes? So in a fearful facial expression, if we were able to take the face away and just leave eyes, is that enough information to capture your attention and you orient towards that location? So the way that we did this, we used a normal dot probe task. We had the fearful eyes, and then we had scrambled eyes that were the same shape, same amount of white, same amount of black, but they're meaningless because they're scrambled. We also did this with neutral eyes, which we found the same result, but with neutral eyes you could have the argument that fearful has more white, so you're going to tend to the more white part. So then we did this to kind of solidify that it was the fact that it's fearful eyes. We We also did it with where awareness was restricted using backward masking. And what we found, this white line right here is the baseline trial. So congruent was significantly faster than baseline. Incongruent was significantly slower than baseline. So what this means is that if this is faster, that means your attention is oriented towards that side, so you have a faster response. And with this being slower, that means it took that much longer for you to disengage from that location and reorient to the other side of the screen. And it holds true whether the participants were aware of the fearful eyes or were not aware of the fearful eyes. So why are the eyes important? Is it because of the gaze? Because you want to know where the individual is looking. So you look at these guys, you might want to know where they're looking because they all look pretty scared. You notice my Monty Python kid. So this is the task that we had. We had a face show up in the center of the screen. We either had them look to the left or the right, or straight forward. And then a dot would appear either on the left or the right. In this case, it's a congruent trial because the eyes are looking towards the dot. If the eyes were looking the other way, it would be incongruent, or forward would be kind of like our baseline. We also did neutral faces with congruent, incongruent, forward. What we found is that independent of the facial expression, congruent trials were faster than incongruent trials, and forward was faster than incongruent trials. So what this means is, regardless of the facial expression, you orient towards where the eyes are looking. And this is the only one where the expression was significantly different. So what that means is that when you have a fearful face, the way... Well, music downstairs is really distracting. So basically what this means is that when you see fearful eyes, you reorient your attention to the location that much faster than if they're neutral eyes, because neutral eyes would mean that there's not something that's like an immediate threat, because it's just a normal face. So then we asked, well, what is the time course of the capture and hold of visual-spatial attention by fearful faces? To test this, we had two different experiments. We had one experiment where the faces appeared for 133 milliseconds, and then we delayed the amount of time the dot appeared after the faces disappeared. We either had a 0 millisecond, 133 millisecond, or 399 millisecond delay. And then in experiment 2, the faces appeared for 50 milliseconds, and then the dots appeared either 34 milliseconds, 118 milliseconds, 286 milliseconds, or 622 milliseconds after the faces disappeared. That's kind of a mouthful. Don't worry about why the numbers seem arbitrary. There's a reason for it. I'm not going to explain it, but there is a reason why there's random numbers. So, same type of task. You have the dot probe, faces, and then the delay, and then the dot. What we found in experiment 1 is a 0 millisecond delay. Congruent was significantly faster than incongruent. In the 133 millisecond delay, congruent was significantly faster than incongruent, and there was no difference in the 399 millisecond delay. The reason we did experiment 2 is because you noticed that the 0 millisecond delay had a really long time of response. What we think happened is we randomized these different delays one after another, so more times than not there was a delay to the dot. So we think that participants were being caught off guard because they were getting used to the fact that there was a delay to the dot, so maybe they were just kind of surprised and it took them a few milliseconds longer. This graph is also from experiment 1. This is just showing the baseline, so you can see that there is a capture of attention or orienting effect, and also a disengagement effect for both of these trials. So in experiment 2, the other one, they're around 420 milliseconds, so these are lower, so the scaling on the graph is different. But This group was significantly different, so there was an attention effect. There was an attention effect towards the fearful faces, as well with this group, or this condition, I should say. But these two were not significantly different. And then you can see the orienting and disengagement effect at those times as well. So what all that means is that if you see an attention effect up until 286 milliseconds, somewhere in between here, your attention goes back to the middle or somewhere else. So a fearful face can only hold that attention there for less than 286 milliseconds. Somewhere, I don't know where the number is. So going back to backward masking and restricted awareness, it doesn't work for everybody. Some people are better at identifying the fearful face than others. The way that we test this is through a dot probe task. But instead of a dot at the end, we just ask them, was the fearful face on the left or the right, or was there no fearful face at all? And we wanted to see if there's difference in some areas in the insula of the brain, the structure of the insula. See if there's gray matter volume differences in that structure to people that have higher awareness or lower awareness. The reason we picked the insula is because there's been other research, like one research study showed that individuals that are more aware of their own heartbeat actually have greater gray matter volume of their insula. So we wanted to see if this would hold true to perceptual awareness as well. Because there were 3 answer choices, this is the detection accuracy. By chance level, it would be 33%, which is this white line, which also happens to be the mean of all of our subjects. So nobody was significantly above or below the mean. Everyone was pretty close to just chance level of getting something right. And this is the gray matter volume. So even within this small variability of accuracy, we can still see that there's a positive correlation of gray matter volume in the insula. And that's after we control for age and whole brain gray matter volume. This is where the insula is located, right here. This is a horizontal cut. This is the front of the brain, the back of the brain, and you're looking at it from the top. These are just slices that are layered on top of each other, so this is more up towards the bottom. This is more towards the top. And as you can see, it's more of the right insula that has a difference in gray matter. The yellow just means that that area holds true to that correlation. So what does this all mean? Fearful faces capture your spatial attention. The eyes are a really important part of the fearful face, and you do not need to be aware that you saw a fearful face to have a behavioral response to that fearful face. So my real-world example of everything I just said, you see fearful faces, so you look to where they're looking, and you end up seeing the most cruel, I'm trying to quote the movie, the most foul, cruel, bad-tempered rodent you'd ever set your eyes on. So your response is to run away. That's it. Yes?
Where do you get the pictures of the fearful faces?
It's from a database. I don't know what the database is called, but we cite GER is the guy's last name. So it is a standardized database, and I think it's from like Sweden or something.
This one's University of Pennsylvania.
University of Pennsylvania. Sorry, the other one's from
Sweden. People
Sweden.
who are like pretending to be afraid, or are they actually afraid?
I don't really know.
Seems kind of key.
Yeah.
Thank you.
Yes.
Was there any experiments to test gender? Like if certain gender, male or female, have different reactions to faces?
I am not aware of that, but I do. This is kind of different from what you're saying.
Yeah.
But I do know that there's a study that they did in Atlanta, where in Atlanta, around Emory and Georgia State is typically African American population. And they found that they have a greater attentional bias towards white faces in general than they do of their own race. And it holds true to white people have a greater attentional bias towards black people's faces as well. And I also kind of wonder if it's just like what you're, like around here there's mostly white people. So maybe it's because like around here we're used to just seeing those faces, and in Georgia and Atlanta they're not used to seeing white people's faces. So I'm wondering if it's just more of like what you're used to. So like if you were a white person that grew up in like a predominantly African American area, if you would exhibit that attentional bias. But as for gender, I am not aware of that.
Okay, thanks.
Mm-hmm. Yes.
Is this? Do you think this is a learned response? I mean, if you tested little children, and then
Little kids typically, I mean, it's kind of hard because they have a very short attention span anyways. But they typically have a response to any exaggerated face. So it's kind of hard to say if they have a bias towards fearful face because it's true, but they also have a bias towards happy or angry, or which we also have an attentional, even adults have an attentional bias towards angry faces as well. Because that's part of the threat response, because an angry face is a threat towards you, where a fearful face is just a threat somewhere around you.
very interesting.
Yeah. That's just to advertise the next people. Any others? Yes. Yeah.
Yeah, has anybody ever done work with primates on fear recognition?
Say that again?
Has anybody ever done work with primates on fear recognition? Fearful face
No.
recognition?
You know that answer?
Yeah. I mean, I think you see the same response.
He says yes, they think they'll see the same response.
Primates.
Yeah.
Interesting. With other primates or with humans?
I don't know. I mean, I guess I think maybe because if you think of like the eyes of primates and humans, like we are primates, so it's very similar like face.
Great.
And I would even think like a dog, if just having those fearful eyes would capture your attention. I don't know of any research that has done that, but it seems to make logical sense.
Sure, because if you think of like dogs, are very sensitive to human fear.
Mm-hmm.
And it's like, it's like, how do they know that? They smell it, or
Yeah.
how do they know it? But they also
It could be your eyes.
could be.
Jake, you had a question?
Has there been research like you did with the eyes of other features of fearful faces? Just isolate the one feature?
Not that I know.
Okay.
All right. Well, if anyone does have any more questions and doesn't want to speak up, come talk to me. I'll be around drinking beer.